I'm John Lunger, Chief Patient Supply Officer at Adaptimmune, and I'll be taking you through some recent highlights and future plans as we continue to work to design and deliver cell therapies for people with cancer. I would refer you to the disclaimers regarding forward-looking statements outlined here. At our Investor Day in November last year, we laid out our 5-year strategic plan for the company, which we refer to as our 2252 plan. We are making good progress against each of these, with the most recent updates being, we are working hard to file a BLA for our first generation SPEAR T-cell therapy targeting MAGE-A4 called afami-cel next year for people with synovial sarcoma. The SURPASS trial data update we presented at ESMO showing clinical responses across 5 solid tumors and an overall response rate of 36% in evaluable patients treated with our next gen therapy, ADP A2 M4 CD8. The start of SURPASS-2, a phase 2 trial investigating ADP A2 M4 CD8 in esophageal and EGJ cancers, which, pending results, could become our second pivotal trial after SPEARHEAD-1. Our collaboration agreement with Genentech, and our intent to file the IND for a MAGE-A4 targeted allogeneic therapy by late 2023. Before I cover some of these recent updates, I want to say a few words about our integrated cell therapy capabilities. We believe that being an integrated company is a crucial factor to delivering these value drivers into our success in meeting our mission to transform the lives of people with cancer. To make the most of the opportunities and tackle the challenges of cell therapy requires a particular set of capabilities. We have built Adaptimmune from the ground up with the sole purpose to design, develop, and deliver cell therapies. Integrating these capabilities, largely internally, enables us to operate faster, better, and cheaper by communicating insights from one part of the organization to every other part quickly and in an iterative fashion. I will now take you through our late and early stage pipeline. Let's start with our MAGE-A4 targeted therapies. First, afami-cel, our most advanced therapies, for which we intend to file a BLA next year for people living with synovial sarcoma. And next, our next gen therapy, ADP A2 M4 CD8, which I've mentioned a few moments ago, being investigated in SURPASS, a phase 1 trial in 5 solid tumors, and SURPASS-2, a phase 2 trial in esophageal and EGJ cancers. I will share some of the data supporting both products in a moment. The third product in our late stage pipeline is ADP A2 AFP. We recently reported data from the phase 1 trial showing encouraging and significant anti-tumor activity with this first generation product. It is encouraging for the potential of cell therapy for the treatment of liver cancer in these heavily pretreated patients with late stage disease. We are continuing to treat patients in the expansion phase and will update when new data are available. Looking at our preclinical autologous pipeline, I will not cover everything in detail, but I want to point out a few specifics. Our autologous products that could enter the clinic include a range of next generation products targeting MAGE-A4 and AFP, including in-house constructs and those developed in partnerships, for example, with Noilimmune's IL-7 CCL19 construct. With our SPEAR T cells currently targeting antigens in the context of HLA-A2, we are also working to expand the HLA populations we reach. Finally, we are also expanding into additional types of autologous cell therapies, including HLA-independent TCRs, or HITs, to do away with the need for HLA typing, and next-gen tumor-infiltrating lymphocytes, or TILs, for which in collaboration with Denmark's CCIT, we will add our next-gen IL-7 to their TIL therapy. And finally, this is our preclinical allogeneic pipeline. Our first allogeneic product in the clinic will be a MAGE-A4 targeted therapy, and we aim to file the IND in late 2023. In our partnership with Astellas, The first nominated target is mesothelin, and we are using our HIT platform. We expect that this will be our second allogeneic product in the clinic. Work on the second target with Astellas is also underway. And we will be working with Genentech to plug their TCR technology into our stem cell platform to create a range of new off-the-shelf TCR products and a novel personalized product platform. I will now give you more details about some of our recent clinical updates. This is the Spearhead-1 data that we reported at ASCO earlier this year. Spearhead-1 is the phase 2 trial with our first generation afami-cel for people with advanced synovial sarcoma. These are best overall response data for the 33 patients in Spearhead-1 who had at least one scan at the time of the data cutoff. The overall response rate was 39% and 41% for patients with synovial sarcoma, including 2 patients with complete responses. There was also 1 PR and 2 stable disease out of the 4 patients with MRCLS denoted by the asterisk. There was an overall disease control rate of 85%. Although these data are early, these results are considerably superior to currently available second-line therapies in synovial sarcoma. We will use this data to file a BLA next year. With ADP A2M4 CD8, our first next-generation therapy, we intended to make a more potent next-generation MAGE-A4 targeted product by adding a CD8 alpha co-receptor to the engineered MAGE-A4 TCR. We started the phase 1 SURPASS to investigate the safety and efficacy of ADP A2M4 CD8, focusing on enrolling patients with advanced gastric, esophageal, esophagogastric junction, bladder, lung, head and neck, and ovarian cancers. These data, reported at ESMO a few weeks ago, are best overall response data for the 22 patients in the SURPASS trial who had at least one evaluable scan at the time of the data cutoff. We have clinical responses in 5 different solid tumor indications, an overall response rate of 36%, and a complete response in a patient with ovarian cancer. You can see from the waterfall plot that the majority of patients experienced tumor reduction and the disease control rate was 86%, which is a great result given the patients in our trial are heavily pretreated and all have very advanced cancers. As you can see, in both Spearhead-1 and SURPASS, most adverse events were consistent with those typically experienced by cancer patients undergoing cytotoxic chemotherapy and/or adoptive cell therapy. In addition, adverse events reported in the long-term follow-up showed no evidence of on-target off-tumor toxicity. Overall, the safety profile to date is associated with an acceptable risk-benefit balance for the indications under investigation for both afami-cel and ADP A2M4 CD8. Taking an in-depth look at each indication where we have seen responses to date with our MAGE-A4 targeted SPEAR T cells. The total potential MAGE-A4 positive population is around 95,000 patients per year based on mortality data. Of those, we estimate around 40,000 also express HLA-A2. We believe that this is only the beginning for our MAGE-A4 franchise, as we are working on further next-gen approaches and broader HLA candidates. In addition, all of these therapies can ultimately be used in our allogeneic program, which I will briefly discuss next. A key element of our platform is that we use a single source of cells, meaning all of the cells come from one original donor. Some features of the induced pluripotent stem cells made from umbilical cord blood that we use include those cells were reprogrammed back to a completely pluripotent state, and a single cell clones were isolated to generate a starting line. Cells that are in a truly pluripotent state have the potential to turn into any cell type found in the human body if they are given the right cues and signals to differentiate down the relevant developmental pathways. They have a very high proliferative potential, Theoretically, they can expand indefinitely in the pluripotent state, allowing us to make very large banks of those cells and giving us reproducible starting material for our platform. The capacity for expansion and ability to bank the cells also allows for a very flexible platform. We are using gene editing, allowing us to do multiple different edits, either sequentially or in parallel, so that we can modify multiple genes. Using editing technology means that we don't have issues with lentiviral vector capacity limits that could restrict the changes we can make. Editing gives us the flexibility to build our future programs however we like based on translational learnings. Finally, we believe that this is a scalable platform. Because we use a single source, there is a single line to fully characterize for each product. Our platform uses defined media as we go through the differentiation process to make T cells from the edited stem cells. We don't use serum and we don't use feeder cell lines. These are both very important factors to enable GMP manufacturing to work in a sustainable, controlled way. I have covered some of the progress that we have delivered throughout the year. We are well positioned to make a difference for people with cancer, starting with synovial sarcoma as we prepare to file our first BLA for this indication next year. With SURPASS, we have now shown that ADP, A2, M4, CD8 has the potential to bring meaningful benefits to people with cancer across a range of indications. We also have a deep preclinical pipeline with our autologous and allogeneic platform programs and are looking forward to sharing further updates by the end of the year and in 2022. Thank you for your time.